let me know chat if I'm audible and visible to all of you students. Hello, 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 hello. Good evening, all of you students. Good evening, good evening, good evening. Hello, hello, hello. Yes. आज कार्बोहाइड्रेट्स होगा आज. उसके साथ में कार्बोहाइड्रेट्स के साथ we'll also be doing proteins also. एक प्रोटीन में क्या होता है? प्रोटीन से there are different structures of protein, primary structure, secondary structure, tertiary, quaternary structure of protein. ये सारा का सारा information आज होगा. Okay students. So students, if I am audible and visible, quickly tell me students in the chat if I'm audible and visible to all of you students. Hello, hello, hello students, if I'm audible and visible. Students, quickly in the chat if I'm audible and visible to all of you. Hello, hello students, good evening all of you, good evening, good evening. Good evening all of you students. Quickly let me know in the chat right now if I'm audible and visible to all of you students. मैं एक बार फोन में चेक कर लूंगा एक बार. One second students, one second, if I'm audible and visible. All of you students, yes I am audible. Perfect, 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 perfect. Nice. Yes students, I am audible and visible to all of you students. Nice, nice, nice. Okay. Now students, in the first class we did something called as what? In the first class we did something called as your entire amino acid structure. Yes or no, all of you students. Then in the next class, we finished entire your DNA structure, lipids, what is a glycerol, what is a fatty acid. ये fatty acid, glycerol सब हमने पिछले क्लास में पढ़ा था हमने, ठीक है? Now today's main agenda is to teach you something called as carbohydrates as well as to teach you something called as the protein structure. ठीक है? I'll be also teaching you about proteins today. Okay students. Now before we go to carbohydrates students, now do all of you know what is turmeric? Students, do all of you know what is turmeric? Students, do all of you know what is turmeric? All of you in the chat? Yes or no students? Do all of you know what is turmeric? Good evening all of you students. Okay. Do all of you know students what is cinnamon? Cinnamon. Okay. Now cinnamon is also a part of your plant on the outer part. Okay. Now students, I want all of you to realize when a plant is growing, okay, जब एक plant grow कर रहा है ना, it will release certain types of metabolites. Metabolites are molecules produced as a result of metabolism. When a plant does metabolism, all of you know what is metabolism, anabolism, catabolism, right? So when a plant is doing metabolism, it is releasing certain metabolites. Okay. Now students, I want all of you to realize some metabolites are called as primary metabolites like carbohydrates, lipids, proteins, nucleic acids. These are called as primary metabolites. Now these are used by the plant for growth. Carbohydrates, lipids, proteins are used by the plants for growth. Now a same plant can also release something called as secondary metabolites. Students, all of you know what is rubber? Nidhi ma'am calls me Abu. Okay, Abu, do you know what is rubber? Aditya, do you know what is rubber? So you might know what is rubber also. Students, have you noticed that एक plant के tree के side, students, एक tree के बगल में ना, you might have seen some gum-like structure coming out. Gum-like structure. Students, have you seen that yellow color resin-like structure? Resin, you might have seen resin. Do all of you know what is resin? Students, I don't know what you call resin in Hindi. Students, what do you call resin in Hindi, students? Can anyone tell me? Do you know what do you call resin in Hindi? Students, have you seen this resin-like structure from a tree? Kishmish. Students, have you seen this near a tree? Tree के पास होता है ना ऐसा, इस तरह का, like yellow color का, yellow. Students, you might have seen near a tree. These are called as resin. Okay. Now all of these resins, all of these gums, right? All of them are called as secondary metabolite. Now students, what is the purpose of, right? What purpose? Not no. Resin is different. That is raisin. That is kishmish is different. Right? What different? Okay. Now students, these are called as secondary metabolites. Secondary metabolites are produced for defense purposes, which we use. Okay. Now there are antibodies, gums, essential oils. Students, you might have some plants have very beautiful smell. No, if you cut the plant a little bit, it will release some beautiful smell. That is called essential oils. right? Scents, spices, right? All of these are called as the secondary metabolites. Okay. Similar students, similarly, there's a list here which I want all of you to see. Okay. Now there is something called as different types of secondary metabolites and types of it. Now, for example, students, how many of you know what is vinca rosa? Students, crack हो रहा है क्या? Okay. Students, how many of you know what is vinca rosa plant? देखा कभी तुमने vinca rosa? Students, how many of you seen vinca rosa? Students, vinca rosa plant. Let me just show you. Let me just show you. Vinca rosa. Students, I don't know what is it called. It's called vinca rosa only. Students, ये देखा क्या तुमने अपने घर के पास? Like बहुत famous plant है, बहुत घरों के पास रहता है. Students, have you seen these plants? They come in white and they come in pink. These are called as vinca rosa. These are called as vinca rosa, students. These plants actually have anti-cancerous property. Yes, they do. They have anti-cancerous property. They can cure cancer actually to some extent. They can control the cancer to some extent. The control of the growth of the tumor. They can do it. Okay, because these vinca rosa, they have something called as, students, vinblastin. They have vinblastin. So vinblastin is a drug. Vinblastin is a drug which is derived from vinca rosa. Very common plant which all of you see. Okay. Then there is curcumin. Curcumin is from your haldi. Curcumin longa. Right. Curcumin is obtained from your entire haldi. Haldi se milta hai curcumin. Okay. Then there are toxins, essential oils. All of you know the lemongrass oil, bath ke dalte na, different essential oils. Then alkaloids, pigments, and drugs and polymer substances like gums, cellulose, rubber. The cellulose which we obtain for making the paper. Paper is made from cellulose, right? All of you know. Paper is obtained from plants, but plant has something called as cellulose. Okay, plant has something called as cellulose, from which we make the paper also. Okay, so these are called as your secondary metabolites. Sometimes they can ask you in examination, what is secondary metabolites, students? Secondary metabolites are the metabolites produced by the plants for what? For defense mechanism. Dishum karne ke liye. Okay, for defense mechanism. That is called as your secondary metabolites, students. Now we come to carbohydrates. Students, now we come to carbohydrates. Ready all of you, students? Ready all of you to understand? You don't have to learn all of them. Just know what is secondary metabolite. Just know what is secondary metabolite and some idea you should have. Okay, you don't have to by heart anything like such. Okay, students. Now, shall we start, students? Shall we start carbohydrates? All of you ready? All of you ready, students? New topic for all of you, students. New topic for all of you. Okay. So, students, I want all of you to understand carbohydrates are nothing but hydrates of carbon and hydrates of carbon. Now, what do I mean by hydrates of carbon, students? Carbohydrates are mainly made up of two different elements, right? They are made up of something called as carbon as a backbone, as well as they have the hydrogen also as a backbone here. So these are called as your hydrocarbons. Okay, students, you might have seen them, right? Like carbon, hydrogen. These are called as your carbohydrates. Okay, students, you know something called as your glucose. Glucose is a carbohydrate. Okay, so today I'll be teaching you what is monosaccharide, what is disaccharide, what is oligosaccharide, and what is polysaccharide. Very simple, students. Very simple it is. Okay. Now, students, I want all of you to understand carbohydrates can be of multiple types. A carbohydrate can be mono, di, oligo, polysaccharide. Now, students, what is a monosaccharide? Understand. What is a monosaccharide? What is a monosaccharide, students? Okay. Now, it is the simplest carbohydrate. Very simple. Okay. It forms the building block of largely carbohydrates. It cannot be hydrolyzed into small units. Let's say triose is an example. You cannot break triose into more smaller fragment, and these are made up of a single monomeric unit. Okay, see, only one unit. So monosaccharides are made up of one monomer. This is a one monomer. There is not one more monomer is not joining here. It has a single monomer here which is independent. You cannot divide it further. Like, you know, it is the smallest thing. It cannot divide further. You cannot break it more. You cannot break it more at all. That is a monosaccharide, students. See, monosaccharide, these are the few examples: pentose, hexose, right? Tetrose and triose. They have 3 carbon, 4 carbon, 5 carbon, 6 carbon, but they are called as monosaccharide. They are the simplest type of carbohydrates. Okay. Now, students, then what is a disaccharide, students? First of all, understand, di means what? 2. Saccharide basically means sugar. Okay. Here, in the case of disaccharides, we have 2 monomer units joined together with the help of glycosidic bond. See, students, see this. For example, here is one monosaccharide. These two are joined together, we get a disaccharide. So when there are 2 monosaccharides joined together, we get a disaccharide. You can learn the structure of glucose, that is more than enough. Structure of a glucose. Okay. All you students just learn the structure of C6H12O6, more than enough, nothing more than that. Okay. This is the structure here. Okay. Now, in examination, generally they'll not ask you to draw the structure, or they'll not ask you to identify the structure also. But in examination, what they will ask you? In examination, आपको पूछेंगे वो लोग कि what are the examples of disaccharide, or they'll give you multiple examples. You're supposed to identify which one is a disaccharide and what is disaccharide made up of. For example, right? Students, understand. Understand. In question, what they'll ask you? In question, they can ask you is what is sucrose and lactose made up of? That is a disaccharide. See, these two are examples of disaccharides. Now that's what why question. This is the why question here. Okay, these are called as your disaccharides. Now the question they'll ask you is what are they made up of? Okay, now sucrose is made up of glucose plus fructose. Glucose and fructose, both of them are what? Both of them are monosaccharide. This is a monosaccharide. This is a monosaccharide. Both of them are monosaccharides. These monosaccharides are joined together. When you take a glucose, when you take a fructose, if you join together, we get a sucrose. So students, sucrose is made up of what? Sucrose is made up of glucose plus fructose. इतना याद रहेगा सबको? Students, इतना याद रहेगा students, इतना रहेगा सबको? Yes, see, I'll tell you what structure they can generally ask you. They can ask you this structure here. They can ask you this structure here, or if they ask you this structure also, okay, I'll tell you how to identify. Students, I'll tell you how to identify. Okay. Now, basic way to difference between, if they ask you the difference between glucose and fructose, just try to look at the group present here. If you can find CHO group anywhere, CHO group in a structure, then it is automatically glucose. Glucose will always have a aldehyde group to it. Okay. Now, instead of CHO, friends, let's say you find C double bond O somewhere in the middle. In the structure, if you find C double bond O, then it is a ketone group. So, ketone group is present where? In the case of fructose. That is a basic difference between glucose and fructose. Students, see here, how many carbon? 1, 2, 3, 4, 5, 6. 6 carbon. 1, 2, 3, 4, 5, 6 carbon. So both glucose and fructose, both are 6 carbon. दोनों के अंदर 6 carbon नहीं है, 6 carbon है. But difference is the functional group here. Here we can see a aldehyde CHO in the case of your glucose. In the case of fructose, we can see a ketone group. Okay, students, is that clear to all of you? Students, is that clear? Difference is clear to all of you. Difference सबको याद रखना चाहिए. Glucose has aldehyde, fructose has ketone. Students, is that clear to all of you? I want everyone to respond. Students, quickly respond to the chat, all of you. Is that clear to all of you, students? Clear है क्या नहीं जल्दी से बताओ. All students, quickly smash the like button. Students, is that clear to all of you? Glucose के अंदर aldehyde होता है और fructose के अंदर ketone होता है. That is the main structural difference between glucose and fructose. Okay? Perfect, friends. Perfect. Now, students. Now, here I told you, students. Students, can anyone? What is sucrose made up of? Sucrose is made up of, students. Sucrose. Students, bond line you will study in chemistry. Aditya, what is bond line? There you will see something called as your dotted line structure. You will see something called as your square structure. All that you learn in chemistry. You learn there is a chapter called as chemical coordination, chemical bonding. You learn all that in bonding. Okay, this they'll not ask you more than bond lines. Structure this. Main questions to general students. Can you see here? This is a structure of your glucose here. Okay, so one carbon, second, third, fourth, fifth, sixth. 6 carbons here together. Okay, 6 carbons here together, students. That's all. Okay, so they'll not ask you more than that. What you should remember is what is sucrose made up of and what is lactose made up of. Okay, let me just check that. Let me just check Tatva questions. We'll bring it next class. Students, I'll bring Tatva questions next class. Okay, I'll try to bring Tatva questions next class and I'll explain you what type of question will come. Okay, don't worry, students. Don't worry. Students, whatever I'm teaching is top level. Whatever more than this, they generally don't ask you. If they ask you also, I will try to make sure in the question practice, I'll make sure you solve. Okay, in question practice, I'll make sure you solve them also. Okay, now what you're talking about, alpha structure, the beta structure, right? In Tattva, they might have given, but what is the alpha structure here? What is the beta linkage, alpha 1,4, beta 1,4, beta 1,6? That you generally study in chemistry part. Okay, alpha 1,4 linkage between your two monosaccharides, beta linkage in the case of cellulose. That generally you study in chemistry part. Okay, what is alpha linkage, beta linkage? That you study in chemistry part of biomolecules, not in biology part. Okay, now what is lactose made up of? Lactose is made up of glucose and galactose. So इतना याद students, इतना याद रहेगा ना, इतना याद रहेगा ना. What is sucrose made up of? What is lactose made up of? Okay, sucrose is made up of glucose plus fructose. Lactose is made up of glucose plus galactose. ठीक है students? इतना याद रहेगा सबको? Okay. Now students, what is a oligosaccharide? Understand students? Oligosaccharides are polymers. They are type of polymers only, but they have around 3 to 9 monomers. Okay, let's say monomer, like similarly they can have 3 or they can have 9, up to 9. That is students, that is called as your oligosaccharides. See, in the case of disaccharides, only 2. In the case of disaccharides, only 2 units. See, 2 units, 2 units. Glucose, fructose, glucose, galactose, 2 units. But in the case of oligosaccharides, they can have around 3 to 9 long chain of monosaccharides. Long chain of monosaccharides, students. Okay. Now here are a few examples here, students. Raffinose is an example here. Raffinose is made up of glucose, galactose, and fructose. See, 3 monomers. 1 monomer, second monomer, third monomer. यहाँ पर 3 monomer जुड़ के हमको raffinose मिलेगा. 3 monomer जुड़ के. Okay. Now one more example is stylose here. Again, glucose, galactose, fructose. 4 monomeric units. So similarly, oligosaccharides में बहुत सारे examples हैं. Oligosaccharides तो पूछते नहीं है. This you can remember for oligosaccharide. Raffinose you can remember. Only raffinose. All of you can remember raffinose. ठीक है? That is glucose, galactose, fructose. Students, same है almost. देखो, इस glucose plus galactose में अगर मैं fructose add कर दूं, it becomes raffinose. Is glucose fructose में if I add galactose, it becomes raffinose. Okay, it becomes raffinose here. It becomes raffinose. That is the oligosaccharide. Then students, what is a polysaccharide then? Should understand. Polysaccharides are your, poly means what, students? Poly means many. Poly means many. Here we can find many sugar units. Many sugar units. Like बहुत बड़ा line है यहाँ पर. बहुत ही बड़ा line है यहाँ पर. Okay, बहुत ही बड़ा line है in the case of polysaccharide, students. Okay. Now students, I want all of you to understand. यहाँ पर ना polysaccharides में based on structure we have different types. Based on storage we have different types. Okay. Now for example, see this, students. See this. For storage, here we have. For structure, here we have, students. These are called, students. I want all of you to understand. Polysaccharides, okay, they can be broadly classified into homopolysaccharides and heteropolysaccharides. Okay, we have homopolysaccharides and heteropolysaccharides. Now, what is the difference? Students, understand here. In the case of homo, students, what homo means same. यहां पर बहुत सारे मोनोमर्स लाइन में जुड़े हुए हैं और ये सारे के सारे मोनोमर्स आर सेम मोनोमर. फॉर एग्जाम्पल, राइट, Karthik, you listen to my class. Okay, in the case of homopolysaccharide, let's say in example, I'll tell you example in homopolysaccharide हमारे पास बहुत सारे glucose, 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 glucose. Imagine a chain, students. Imagine a chain like this: glucose, 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 glucose. See, glucose is a monomer. ठीक है? Glucose is a monomer. Okay, glucose is a monomer. Glucose is a monomer. But when one long chain of glucose, that is a polymer. But if it is made up of only glucose, यहाँ पर कुछ extra नहीं है, यहाँ पर सिर्फ glucose है, that is a homopolysaccharide. Students, एक सिंपल समझो, तुम्हारी क्लास की एक लाइन खड़ी है. Students, do all of you stand in one line in students assembly में खड़े होते हो तुम लोग? Students, do all of you stand in assembly in one line? Yes or no? All of you belong to the same class. एक 9th की क्लास है, एक 8th की, 6th की, 5th की क्लास है. Boys girls separate है, ठीक है, वो तो ठीक है. But you have all 9th in one group, all 9th here, all 10th, all 11th. That is a homopolysaccharide. Okay, all of them belong to same class. Homopolysaccharide. Okay, these are examples of homopolysaccharides. Examples of homopolysaccharides here. We have starch, glycogen, inulin, cellulose, chitin. Now, what are they made up of? What is the monomer here? In the case of starch and glycogen, the monomer is glucose. Both starch and glycogen, they are made up of long chain of glucose. Students, is that clear to all of you? Students, is that clear to all of you? In the case of starch and glycogen, they are made up of long chain of glucose. Okay, see, starch के अंदर starch के अंदर glucose. This is how starch looks like. Students, starch will also show some branching also like this. Branching. This is how starch looks. Okay, this is how the starch looks like. Okay, and students, remember, starch is food reserve in the case of plants, consists of amylopectin and amylose. These two are different types of starch. Amylopectin and amylose, students, amylopectin and amylose are two different types of starch here. Amylose and amylopectin are two types of starch. Okay, now glycogen, it is again a polymer of glucose. But students, glycogen is a food reserved material in the case of animals. Now you might be asking me, sir, is glycogen and starch same to same? Students, do you think they are same? Students, do you think they are same to same? All of you? Do you think starch and glycogen are same? Because both are polysaccharides, both polymer is glucose only. But do you think they are same to same? All of you? Students, do you think they are same? No. Why are they not same, students? Why are they not same? Let me tell you, students. Let me tell you. Here we have, let's take starch here, and we can see glycogen here. Okay, glucose. 1 2 3 4 5 6. 1 2 3 4 5 6. Okay, now you know the difference, students. One glucose. This is called a side chain. Okay, Side chain is here. Now students, in the case of glycogen, what will be there students? In the case of glycogen, this is called as alpha 1,6 linkage on the side. In the case of glycogen, glucose. Students, do you see the difference here? Students, do all of you see the difference here? Students, do you all of you see the difference? Abu, do you see the difference here? In the case of starch, the branching is not very frequent. In the case of starch, the branching is not very frequent. Very rarely the branching happens. But in the case of your glycogen, long chain. See, I have drawn here only very few, but it is a very long chain. But students, here you can see here the chain is very frequent here. So that is why we say glycogen is highly branched. Glycogen is highly branched compared to starch. See, starch and glycogen दोनों की दोनों है ना ग्लूकोज से बने, बट यहां पर डिफरेंस क्या पता है? यहां पर ग्लाइकोजन के अंदर वो ब्रांचिंग है ना, ब्रांचिंग बहुत फ्रीक्वेंटली होती है ब्रांचिंग. Students, did all of you understand? Students, did all of you understand? The branching is very frequent in the case of your glycogen compared to starch. That is the main difference. Again, it is found in the case of plants, found in the case of animals. Students, हमारे बॉडी के अंदर do you find glycogen or do you find starch? Students, हमारे बॉडी के अंदर do you find glycogen or do you find starch? Quick answer I want, students. Quick answer. In our body, we get glycogen. We have glycogen, students, in our body. Okay. Now, what is inulin, students? Inulin. Now, inulin is found in the case of plants, but what is the monomer here? In the case of inulin, the monomer, students, is a fructose. Inulin, not insulin. Inulin. Inulin के पास ना long chain of fructose है. Fructose. Long chain of fructose. यहाँ पर ग्लूकोज था, यहाँ पर फ्रक्टोज है. Okay, that is the main difference. So inulin, right? Not insulin. Inulin is different from insulin. Inulin is a polysaccharide. Insulin is a hormone. Insulin is different. It is a hormone. Okay, this is inulin. Then you might get confused. It is very honest confusion. Okay. Then these are called as structural polysaccharide because they help in storage. They give structure. For example, cellulose. All of you know cellulose is used to make paper, to make books, to all of you to study. Okay, it is a polymer of glucose. Again, students, it's a polymer of glucose. Now it is also polymer of glucose, but all of them are same? They are not. Structure of cellulose is very different. Structure of cellulose has something called as beta 1,4 linkage. Let me just show you, students. Let me just show you. You'll get better idea. Students, can all of you see? This is better, easy to understand, students. देखो यहाँ पर भी ग्लूकोज है. Students, यहाँ पर भी ग्लूकोज है, यहाँ पर ग्लूकोज है. Now this is called as beta 1,4 linkage. This linkage is found in the case of your cellulose. This is cellulose structure. Can you see beta 1,4 linkage in the case of cellulose? यहाँ पर भी ग्लूकोज, यहाँ पर भी ग्लूकोज है. Long chain of glucose. हर एक ग्लूकोज के बीच में we have beta 1,4 linkage. Now students, similarly, in the case of, you see, students, दिख रहा है डिफरेंस? डिफरेंस दिख रहा है क्या? Can you see? This is the structure of your starch, right? Sorry, this is the structure of starch. यहाँ पर alpha 1,4 linkage है. This is bottom linkage here. Cellulose के पास top पे था beta 1,4. This is alpha 1,4 linkage. यहाँ पर आपको दिख रहा होगा 1,6. This is called alpha 1,6 linkage. This is the one more branching. This is the branching here. Branching. Okay. Now this branching is very frequent. This branching is very frequent in the case of your glycogen. तो बाहर पटाखे फोड़ रहे स्टूडेंट्स, बाहर पटाखे फोड़ रहे हैं, पता नहीं क्यों. Okay. Now student, this is called as your, can you see, alpha 1,4 linkage in the case of starch. Okay. This is your alpha 1,4 linkage in the case of your starch. That is the main difference. Starch, glucose, fructose, cellulose also made of glucose. But students, here the structure is different here. Here we have structure is different here. I'll teach you what is H2A, what is H2B in the case of hemoglobin. I'll teach you today. Okay. Then student, there is chitin. Students, can anyone tell me where have you studied about chitin? Students, हमने पढ़ा था क्या chitin के बारे में? Students, have you studied about chitin somewhere? It is present in the case of your, anyway students, chitin. Students, chitin is a polymer of N-acetyl glucosamine. Okay, it is present in the arthropods, in fungi also. Yes or no? Right? Chitin is also present in your exoskeleton. This cockroach है ना, cockroach को बाहर जो है ना, बाहर का, that is also made up of chitin. That is also made up of chitin here. Okay, that is exoskeleton of your this thing. Okay, next students, exoskeleton of your arthropods. Okay, students, these are called a that is called as your homopolysaccharides. All of these are examples of homopolysaccharides. Homo, made up of N-acetyl glucosamine, long chain of glucose, fructose, glucose. Then students, we have something called as heteropolysaccharides. Students, सबको याद होगा ये? Students, do all of you remember this? Students, कहाँ पढ़ा था हमने पेप्टिडोग्लाइकन के बारे में? Students, हमने पेप्टिडोग्लाइकन के बारे में कहाँ पढ़ा था? जल्दी से बताइए सब लोग, कहाँ पढ़ा था हमने पेप्टिडोग्लाइकन के बारे में? सेल वॉल के पास बैक्टीरिया. यस, सो बैक्टीरियल सेल वॉल जो है ना पेप्टिडोग्लाइकन, दैट इज अ हेटेरोपॉलीसैकेराइड. दैट इज अ हेटेरोपॉलीसैकेराइड. ठीक है? Do all of you remember? I told you it is made up of two different types of repeating units, NAG and NAM. NAG and NAM. N-acetyl glucosamine, N-acetyl muramic acid. This is your NAM. NAG and NAM. NAG and NAM. Repeating units of NAG and NAM, NAG and NAM. Yes or no? NAG and NAM. That is a heteropolysaccharide. Heteropolysaccharide because NAG, NAM, different units. Different repeating units. One more example is agar-agar, students. Agar-agar is like a substance which is obtained from your algae, red algae, Gelidium and Gracilaria. It is obtained from algae and it is used to make jellies, it is used to make ice creams, it is used in microbiology to make to grow bacteria. It is used there. It is also a type of heteropolysaccharide. It is a type of heteropolysaccharide, students, heteropolysaccharide. So this structure they will not ask you, but examples they can ask you. Heteropolysaccharide examples they can ask you, students. NAG and NAM. Okay. Now students, I'll teach you something what is reducing sugars. This generally you will learn in chemistry, but I'll teach you in small 2-3 lines I'll teach you, students. What do I mean by reducing sugar? Reducing sugar, students. Reducing sugars are the sugars, because sugar is basically carbohydrate, right? A carbohydrate or a sugar which has a free aldehyde and a ketone group. So can you see, students? Like glucose, fructose, galactose, do all of you see they have a free aldehyde, ketone and aldehyde group to it, free aldehyde. Okay, now whenever there is a free aldehyde group, right, this can actually undergo reaction. Okay, so reducing sugars, we have free aldehyde ketone group. Reducing sugars reduce the cupric ions in Benedict solution to cuprous. So let's take one small test tube. We add 2 ml of glucose. To that we add equal amount of Benedict solution. This Benedict solution has a lot of cupric ions in it. Yes, these cupric ions will react with the aldehyde and ketone and it turns into brick red. It turns exactly. in your diabetes. Exactly, students. If you heat in water bath, it becomes brick red. That is, reducing sugar undergo reaction. Reducing sugars are the sugars which have a free aldehyde and a ketone group, students. So all the reducing sugars will have a free aldehyde and a ketone group. They have a free aldehyde and a ketone group. Okay. Now, students, what about your disaccharides like sucrose? They will not, because the aldehyde part undergoes bonding. Can you see, students? It undergoes bonding here. The aldehyde part undergoes bonding, so there is no free aldehyde. There is no free aldehyde here. Free aldehyde is not present, so it doesn't undergo reaction. Okay. Non-reducing sugars, no free aldehyde or ketone group. All polysaccharides and sucrose are non-reducing sugar. So all polysaccharides and sucrose are non-reducing sugar. They will never react. They cannot react with Benedict solution because they do not have a free aldehyde and ketone group. Okay, they do not have a free. Students, one more example, one more test I will teach you, right? What is called as iodine test. Students, you might have heard about this, students. All of you have heard about it, students. Exactly, students. Bechara sucrose. Okay, students, how many of you heard about iodine test recently? Or fake paneer के ऊपर करते ना, fake paneer के ऊपर. Students, you might have heard about fake paneer. Yes or no? Students, how many of you heard about fake paneer? Yes. Iodine test. Okay. Now, students, we will do a similar iodine test right now. Right? Similar iodine test right now based on the plant here. Okay. How in the case of your saccharides. Now, let's take iodine solution and we add starch. Iodine solution and we add cellulose. Now, students, remember I told you. Can anyone remind me, starch and cellulose, which monosaccharide they are made up of? Starch and cellulose, they are made up of which monosaccharide? Can anyone remind me again, students? Can anyone remind me? Anyone? Which monosaccharide they made of, students? Starch, they are made up of glucose. Both starch and cellulose are made up of glucose. Both are polysaccharides. They are both are polysaccharides, and the monomer here is glucose. The monomer. See, students, दिख रहा है ना यहाँ पर? देखो, दिख रहा है ये वाला diagram. यहाँ पर this is a polysaccharide. It is a polysaccharide, but it is made up of single monomer. 1, 2, 3, 4, 5 individual monomers. Students, what is this monomer called as? In the case of your starch, what is that monomer called as, students? Monomer. Individual monomer. Individual monomer is called as glucose. It is made up of glucose only, right? Individual monomer is a glucose there. Here, just go back and I'll show you. Here I told you, students, cellulose is a polymer of glucose. Many glucose joined together, we get cellulose. Starch again is a polymer of glucose. It is a polymer of glucose, students. Yes, sugar which we eat also. Glucose. Sucrose. That what we eat is sucrose. Students, what you eat is sucrose. You don't eat glucose. You generally eat sucrose. All of you generally eat sucrose. Ghar chini hai na, chini. Chini is a sucrose. Chini is called as sucrose. Okay. Chini is called as sucrose, which what you eat. Disaccharides. You all of you eat disaccharides. Okay. Now, students, what exactly happens in this test? You understand, students? In this test, what is happening? When you use iodine on starch and cellulose, in the case of, students, in the case of starch, we get blue color, but in the case of cellulose, we get yellow color. Why? Okay. Yes, fructose is edible. A lot of people eat fructose also. Yes. Now, students, why are we getting different colors? Now, students, let me just give you a small example. Okay. Now, I'll throw a ball to you, and can you catch it? Can you catch the ball? Like, मैंने ball पकड़ा हाथ के अंदर. Forget ball, students. If I take this water bottle and throw the water bottle to you, will you be able to catch the water bottle? All of you tell me yes or no. All of you can catch the water bottle with your hands. You can catch. Now, what if I will tie your hands? Students, I will completely tie your hands. हाथ को बांध दूंगा मैं, ठीक है? उसके बाद मैं बॉटल थ्रो करूंगा. Now, can you catch the ball? Can you catch the bottle now, students? Students, can you catch the bottle now? Not possible, because your hands are tied. If your hands are tied, how can you catch? Not possible. Same concept here also, students. Here, if you look at the structure of starch and structure of cellulose, the structure is very different. Not legs, I'm telling you with hand only. Legs will catch up, one ulta palti will do. So students, if you look at the structure of starch here, you can notice starch has something called as helicals. Can you see? These are called as branching in the case of starch. Branching, can you see students? Branching can be seen here. Branching can be seen here. In the case of amylopectin especially. Amylose not really. Amylopectin you can see the branching here. So starch can hold the iodine molecule. It can hold the iodine molecule. Like how you can hold the bottle with your hands now. So your starch can hold the iodine molecules. Okay, in the helical portion, and that is how it looks blue color. That is how it looks blue color. But students, here in the case of cellulose, it doesn't have any branching. No branching at all. Okay, so cellulose does not contain complex helicals, and it does not hold I2. It cannot hold iodine molecule, and that is why we get yellow color here or brown color. That is the original color of iodine. It comes out directly. Okay, that is the main difference, is the structure difference between cellulose and starch. Okay, students, answers are given by mistake. Students, I hope all of you can answer these questions easily. Students, answers are given. I hope all of you can answer these. These are NEET PYQs. These are actually NEET PYQs. Inulin is a polymer of fructose. Yes or no? Exoskeleton of arthropod is made up of chitin. It is made of chitin, students. It is made up of chitin. Okay. Now, students, there is a very important thing here. Okay. Students, हो जाएगा, protein हो जाएगा. Students, protein में उतना है नहीं. Protein I'll be teaching you structure today. I won't be teaching you more because protein सिखाया मैंने पहले. Proteins are made of amino acids and everything. Okay. Now, students, बहुत simple concept है. समझ लो. They can ask you a question on this. Okay. They can ask you, students, different functions of protein. Different functions of protein. Collagen is a protein. Trypsin. Students, please remember from today onwards, almost all enzymes which we see, bioenzymes, biocatalysts are called as enzymes. All the enzymes are also made up of protein. Please remember that. Many students who are in NEET examination, many students who are in class 12th, they still do not know what are enzymes made up of. Enzymes are made up of proteins. Yes, they are made up of, except the ribozymes, except the RNA enzymes. Ribozyme is an RNA enzyme. Almost all the enzymes are made up of proteins only. Proteins. Okay. So, in hormones are also made up of proteins. Yes. That's why we say eat protein a lot in your diet. That's why we say eat a lot of protein. Your diet should have protein. This is the problem with India. In Indian diet, we hardly include protein. Very less protein is there in our diet. Okay, especially if you're vegetarian, if you don't eat chicken, if you don't eat mutton, don't eat eggs, gone then. The only source of protein which you can have is soya chunks, dal, pulses, paneer, right, or tofu. So please eat a lot of protein in your diet every single day. Every single day, make sure you have some protein in your diet. Okay, now don't go and tell your mother, I want paneer every day. Paneer cannot happen every day. Paneer is very costly. Paneer is very costly, expensive, but don't tell that. At least have some amount of protein in your diet every single day, every single meal actually, every single meal. Thoda dal wahan se yahan dal. Okay, dal yes, eat dal every day. Please eat dal every day. Every day you should eat dal. But वही है, दाल से बहुत कम प्रोटीन मिलता है. So at least try to drink milk every single day. 1 glass of milk, lassi, कुछ भी हो, lassi, milk, drink every day. Curd every single day, curd. ठीक है. दाल चावल, हाँ, दाल चावल वही थोड़ा कम मिलता है दाल से. स्टूडेंट्स, दाल से उतना मिलता नहीं है. ठीक है. सो दाल के साथ ना, try to drink milk, try to eat paneer every single day, almost alternate days, or eat 3 times a day, 3 times a week paneer at least, because we need protein. Eat soya chunks, right? Soya chunks has also a lot of paneer, so a lot of proteins in it, right? That's what answer. Antibodies are also made up of proteins only. Antibodies, protein only. Okay. Receptors, GLUT4 is a type of your protein which helps in transportation, of glucose. Okay. So all of you know your collagen, your ground substance in your body is made of protein. So everything is actually made up of protein only. Okay. And students, here all of you should know collagen. Collagen, students, is actually collagen is the most abundant protein in the animal world. So in the entire animal world, collagen is the most abundant, means most found protein, most found molecule, actually not even protein, most found protein, right? Now if you ask me which is the most abundant protein in the entire biosphere, in the entire world, the most abundant protein in the entire entire world is rubisco, which is an enzyme actually. Is the rubisco. Rubisco is an enzyme which is present in your C3 and C4 cycle. All of you know, right? Students, do you remember C3 cycle, C4 cycle? I have made a video on it, students. I have made a video on it. C3 cycle, C4 cycle. Yes or no? Yes, right. So rubisco is an enzyme. Rubisco is an enzyme which is present everywhere. It is the most abundant protein in the entire biosphere. In animal kingdom, collagen. Animal kingdom, collagen. Biosphere, rubisco. Okay, yes, students. Now we come to the structure of protein. This is the only main part today. After this, class is over. Okay. Students, मैंने पढ़ाया था photosynthesis. हाँ, photosynthesis के अंदर पढ़ाया मैंने वो सारा. Photosynthesis के अंदर पढ़ा है. C3 cycle, C4 cycle, C2 cycle. What is rubisco? What is peptidase enzyme? Yes, I thought all of that in photosynthesis. Okay. Now students, I want all of you to understand the structure of protein. Protein किस तरह से दिखता है? Okay. What is the structure of protein, students? Students, do anyone of you have any idea how does proteins look like? Anyone, any idea? How do you think proteins look like? Students, I want all of you to understand protein is actually a polymer. It is a polymer. Protein is a polymer. Students, what is protein made up of actually? Students, what is protein made up of? H2O, H2B is a type of subunit in right. So what is actually what is the monomer here, students? Can anyone tell me? See, I told you protein is a polymer. Protein एक लंबा polymer है. But what is each individual monomer called as? Each individual monomer is called as an amino acid. It is called as an amino acid, students. So if you take one more amino acids, amino acid, amino acid, amino acids, amino acid. There are 20 amino acids, right? 20 amino acids. You mix and match them. You mix and match all the amino acids, we get the proteins. So from amino acids, if you join them, right, with the help of peptide bond, we get a protein. We get a protein like this, protein. Okay, we get a protein, students. Now, students, I want all of you to understand there is something called as primary structure, secondary structure, quaternary structure, and tertiary structure of protein. Tertiary structure of protein. Okay. Now, this is called as primary structure of protein. In students, do all of you know what is woolen? Woolen, all of you know woolen? Yes or no? All of you know what is woolen? Students, do all of you know what is woolen? Yes or no? Woolen. Okay. Now let's say if I take one, students, what is woolen made up of? Do all of you know what is woolen made up of? Woolen is made up of small thread there, no? Woolen is made up of small thread. Let me just show you. Students, these are called as woolen. All of you know, if you do not know, let me just show you, students. Okay. In winter you wear sweater, no, sweater it is there. Now students, if you see a woolen here, each one strand is made up of small fibers or threads. Now try to imagine one single strand. Students, try to imagine one single strand of woolen here. Okay. Now this single strand of woolen is actually made up of lot of other fibers here. Lot of other fibers. Yes or no? Lot of fibers here. This is a woolen here. Okay. Similarly, students, we have something called as This is the primary structure of protein. Primary structure of protein is like one small thread of woolen. Like here, try to imagine this small thread is like your amino acids. Small threads are like your amino acids. When we have multiple amino acids joining together, we get a protein. Multiple small threads will join together to give you one woolen here. Students, did you understand the analogy here? Students, did all of you understand the analogy here, students? All of you? Yes or no? Did all of you understand? Yes or no? Here, this is a woolen strand. This is a one woolen strand here. One woolen strand. How it is made up of small fibers like this. A protein is also made up of small amino acids. And a single thread like this is actually the primary structure of protein. This is a primary structure of protein. Can you see here? It is made up of amino acids. Amino acids. Okay. Can all of you see here, students? Amino acids 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Like long chain of amino acids, students. Long chain of amino acids. It is like one single thread here. It is like one single thread here. Okay. That is called as primary structure. Now, what about secondary, students? What about secondary structure? Understand? Second structure में क्या हो रहा है पता है? Secondary structure में ना वो thread है ना thread. If I bend the thread and give it a shape. Now, if I bend the thread into somewhat like DNA-like structure, it is called helical structure. It is called as helical structure. Okay. Now, bending the thread. I am bending, students. I am bending the thread here. Can you see? Bending the thread here. Okay. Now, what if I keep the thread next to each other? If I keep the thread next to each other, students, next to each other, that is called as beta plated sheets. So, if you take a primary protein and if you bend it, you can get two different shapes, and these two are called as secondary structure. Alpha helical, beta plated. Okay. Now, students, now what if I take the woolen and I make it round? What do I get, students? What do I get? Let's take, I take this alpha helix and this beta plated. I will take both of these and I'll make a ball out of it, like woolen ball. Yes, so all of you know what is woolen ball? Then we get tertiary structure. Then we get tertiary structure here. Tertiary structure is basically a 3D structure right now. Yes, students, till now the woolen was straight. Woolen was straight like this. But if I take a lot of woolen and I combine it together, I get a ball. This is that ball here. This 3D structure. Then tertiary structure. Now let's say, students, let's say I take one ball here. I take one more ball. 3, 4. I take 4 different types of woolen balls here, students. I will take, sorry, let me just show you, students. This was primary structure. In tertiary, students, in the case of tertiary structure, I am making a ball like this. Tertiary, this is tertiary structure. Now in quaternary, what we do? In the case of quaternary, I take multiple balls like this. I take multiple balls like this and I join them together. I join them together, students. I join them together. This is now quaternary structure of protein. This is now quaternary structure of protein. Now I'll give you a quick example. All of you know the example. Hemoglobin. All of you know what is hemoglobin, students? All of you heard about hemoglobin? Hemoglobin is actually a quaternary structure of protein. Hemoglobin is, this is what hemoglobin looks like. It is a quaternary structure. It has 2 alpha chains and 2 beta chains. Okay, that is your quaternary structure. See, primary structure of protein. Represent the position of amino acids. It tells you which amino acid is present where here. Which amino acid is present where, it tells you. Okay, it has a N terminal, it has C terminal. N terminal because it has a NH2 amino group here. C terminal because it has a carboxylic acid here. So C terminal, N terminal. See, this is alpha helix. Alpha helix structure like a solenoid. Alpha helix structure of protein. This is beta plated. You see beta plated like a paper. This is what protein looks like. Beta plated, students. This is a tertiary structure. In tertiary structure, it forms a ball. It forms a ball suddenly. Ball, tertiary structure, protein. Okay, biologically active. Then, students, we have quaternary structure. In quaternary structure, we have subunits. In students, in quaternary structure, Students, H2A, H2B is part of your next chapter. I've teach that in genetics. That is in genetics. That we learn in genetics actually. In genetics. In genetics, you learn about nucleosomes. You learn about there. There you learn that in the chapter called as molecular basis. Where when I'll teach you something called as your transcription, translation, DNA packaging. Do all of you know what is DNA packaging? There we will learn. And there I'll teach you something called as DNA packaging. We learn that. There is something called nucleosome. Right? There we will learn H2A, H2B. Not here. In this chapter it is not there. In this chapter it is not there, students. In this chapter, right? No, biomolecules will take one more class. In next class, I'll teach you how enzymes work. All of you know what is enzymes. If you do not know what is enzymes, I'll teach you what is enzymes, how do they function, there is something called inhibitors. Enzymes function can be stopped? How does the enzymes work? What is activation energy? Right? What do you mean by your Michaelis constant? That you will learn in the next class. Students, biomolecules is a very big chapter. Biomolecules is a very big chapter, students. Okay, this is your quaternary structure of protein, students. Can you see? Hemoglobin. This is how hemoglobin looks like. Let me just show you one 3D structure of hemoglobin, students. See here. See, this is how proteins look like. First of all, students, this is how proteins look like. Can you see proteins? Okay, students, alpha helix models. Alpha helix models here. When they join together, many alpha helix join together, tertiary, subunit, 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 then becomes quaternary. This is how proteins look like in your body. This is how proteins look like in your body, students. Proteins. Okay, proteins look like this in your body. Now let me just show you hemoglobin, students. Let me show you hemoglobin. This is how hemoglobin looks like. See, this is a hemoglobin. Can you see, students? It has 4 subunits. 4 subunits. 1, 2, 3, 4. 4 subunits are joined together. 2 alpha chains and 2 beta chains here. 2 alpha and 2 beta chains. This is how the actual protein looks like in your body also. In your body, students. Okay. So let me just summarize the entire thing with the help of a small video for all of you. Let me just summarize the entire protein structure with the help of a small video here. Okay. You can see, students, this is a single amino acid. This right here, students, is a single amino acid. All of you know single amino acid. It has a R group, it has a amino group, and it has a carboxylic group. Now, can you see, students? Can all of you see? 2 amino acids join together. When multiple amino acids join together, we get a protein. And the bond here is peptide bond. See, different types of amino acids. Different types of amino acids. Can you see? Different types of amino acids are there. There are 20 different, I told you, there are 20 different amino acids, and these 20 different amino acids will join together to give you a protein. See, this is primary structure. Long chain. Can you see? They are just chain. Long chain. Different amino acids join together. Different amino acids just join together. Okay. Second structure. Okay. Can you, students? Alpha helix, helical structure. They have hydrogen bonding here. This is beta sheets. Beta sheets. Tertiary structure. It becomes a ball right now. Tertiary structure becomes a ball. Can you, students? Tertiary structure becomes like a ball structure. It has an active site also here. It becomes a ball-like structure. Quaternary. Can you see? Multiple join together. In quaternary, we can see multiple subunits joining together. Multiple subunits joining together. Hemoglobin is an example. 4 subunits. It has 4 subunits, students. Okay. Students, that is your entire structure of protein. So, primary structure, secondary has alpha helical and beta plated, tertiary is like a small ball. Then lastly, we have the quaternary structure of protein. Okay, students. So, students, that's it for today's class. That's it for, in next class, students. In next class, I'll be teaching you. In next class, students, I'll be teaching you something called as the function of protein, and this part here, this main part I'll be teaching you. This is the main part I'll be teaching you. What is cat, right? So before I go, students, I want all of you to understand, right? Peptide bond is a bond. Peptide bond is a bond. It is a bond between two amino acids. One amino acid, one more amino acid. Between them, they have peptide bond. Now, what about in the case of polysaccharides? In polysaccharides, the bond is called as glycosidic bond. It's given somewhere, students. Huh? It is called as glycosidic bond. Glycosidic bond is a bond between, students. Glycosidic bond is a bond between two different carbohydrates. One monosaccharide, one more monosaccharide, one more monosaccharide. Okay, so when they join together, that becomes a polysaccharide, and in polysaccharide, this is the bond here, glycosidic bond. Okay, in between carbohydrates, peptide bond in the case of amino, in the protein, that is amino acids. Okay, students. Students, if I teach you right now, I know for sure the brain cannot process, right? I don't mind teaching. I've taken a marathon for 10 hours. I've taken a marathon for 9 hours. I've taken a marathon. Okay. The thing is, the last part of this chapter is the most difficult part of the chapter. If you want to understand the functioning of enzymes, how does the enzyme function, you need a fresh mind. Okay. So in the next class, I'll teach you the entire functioning of the enzymes, and that will be the last class of your biomolecules. Okay, students. So thank you so much, students. I hope today's class was useful to all of you. I hope all of you learned something new today, right? Structure of protein and carbohydrates is done and dusted. It is very easy. Just learn the important terms here and there. What is a homopolysaccharide, heteropolysaccharide, examples of them, and you're good to go, students. So thank you so much, students. Bye bye. Take care, all of you, and I will see you in the next class. Until then, keep studying. Tomorrow I have a special class, students. Tomorrow I have a special class for all of you. Tomorrow I'll be teaching you glycolysis. Okay. Tomorrow I'll be teaching you what is glycolysis. It is the most important process in the human body. Every single organism in the world has to undergo glycolysis. Now, what is glycolysis? You will learn in tomorrow's class. Okay, students. So bye bye, all of you. Take care and have a great day.